Hello, today we are going to learn the experiment 1 with the title Introduction Tools Used in Experiments. Today's contents will cover about the experiment objectives, tools, and their procedures. And then the experiment objectives, the students are hopefully familiar and understand the basic principles of tools used in biochemistry experiments. And the students are expected to use the tools properly during experiments. We will cover 4 common tools used in pharmaceutical biochemistry labs, such as micropipette, pH meter, centrifuge, and SDS-PAGE Electroporator. The first tool that I'm going to explain today is micropipette. Micropipette is a tool used to transfer a small amount of liquid, less than 1 milliliter, with a microliter scale. Usually in a lab, there are 3 types of micropipette with the P1000 scale and P200 scale and P20 scale. As we can see here on the left picture, the micropipette has 6 parts. The first one is a plunger at the top one. The plunger is a button used to eject the liquid, and this plunger has an important information, such as we can see on the middle picture, that the plunger has word P1000, for example. It tells you about the scale of the micropipette. And the second part of the micropipette is a tip eject button. This button is used to unplug the tip at the bottom of the micropipette. And then the third part is a volume adjustment dial. This volume adjustment is used to increase or decrease the volume of liquid. So you just can roll it up to minimum or to maximum one. And then the fourth part is about the volume readout. The volume readout is used to see the maximum volume or the number of volume that we can take it out from the liquid. And then the next part is the tip eject shafts. This is like a thin plate that help the tip eject button to eject or to unplug the tip. And the last part is at the bottom one is the tip attachment. This part is a place for us to put our tip on. And moving on to the right picture, there are 3 types of micropipette here. So P1000 has maximum volume is 1000 microliter or 1 milliliter, and it has the accuracy ranges between 200 and 1000. And while the middle one, the P200, it has a maximum volume is 200 microliter with the accuracy ranges between 20 microliter to 200 microliter. And the last one is the P20 with the maximum volume is 20 microliter, and the accuracy ranges is between 0.5 microliter to 20 microliter. As we can see in the middle volume readout here, for each of the micropipette there are 3 numbers there. Usually they have typically black and red, and the change of the volume adjustment is gonna change the number of the volume here. Like for example, in P1000, the maximum volume at the top of the volume readout box here, it's gonna tells you about 1000 microliter. So if this show you a 9 and 2 and 2, it means that there are 922 microliter. But here, as you can see, that there is 0, 2 and 2, so it means the first one is 1000 point, and the second one is 100 point, and the last one is 10 point. So it's same to the P200 and P20. So As a note, it is prohibited to exceed the upper or lower limit of your micropipette, because if you exceed the upper and lower limit, you can damage the micropipette, or micropipette will be totally broken. So, and be careful of a plunger stop point. So actually the plunger has two stop point. The first one is the first resistance point, so it tells you how many microliter you are going to take out from the sample, and the second one is the last stop point that you can barely push the plunger button. So be careful for the first stop point and the second one. And the next one is be on volume range. Don't go the lower limit or above the upper limit. And for the next one is don't turn the pipette upside down, because if you are working in a sterile condition, they will contaminate your sample. So the next part is if you're going to use the micropipette to take out some liquid, so first press the plunger until it reaches the initial resistance point. So you press for the first stopping point and dip the top of the tip into the liquid just barely below the surface. Remember, just barely below the surface of the liquid, and avoid to touching the bottom of the tip, the bottom of the liquid container. So when as you can see here on the right picture, there is a step-by-step how to use the pipette correctly. So the first is stop point, and the second one you can just blow out all of the sample from the micropipette. So if you are going to use the micropipette, just press the first stop point and put your dip your tip into the sample chamber. And the step 2 is release the plunger slowly until it reach the maximum volume. And the step 3 is taken out the tip slowly from the sample chamber. And the step 4, put it or move the liquid you want to transfer into a new chamber, and then press it slowly until it finish until the liquid is totally transferred into a new place. And release for the step 5, release the plunger also slowly. And then the last step is just push the eject button to unplug the tip. And moving on, for the second tool is a pH meter. pH meter is a scientific instrument that measures the pH or acidity or alkalinity of the buffer solution. This tool, as you can see here on the left picture, it has two important parts. The first one is electrode part that connected to an electronic meter here that can show you that can display the pH reading here. The part of the electrode one is sensitive to H ions and contains AgCl and HCl. If you are going to use the pH meter, the first thing you have to remember that the pH meter have to be calibrated before you use it. So the pH meter calibrated is using 2 or 3 different buffer solution to reach the designated range. For example, you are going to check the sample between the pH 8, so you need 3 different buffer solution between the 4 and the 10, because your pH sample is 8, right? And usually for calibration, they use the ranges pH from 4, 7 and 10. sorry. And usually if you are doing the experiment offline, we need to do some experiment called pH determination. So all you have to do is just determine all of these four different buffer using pH meter, and you can just write it down, the pH measured here on the column pH measure. And moving on to the centrifuge. Centrifuge is a machine to separate particles within a solution due to the gravitational force and a centrifugal force, depending on the particle size, density, and motor speed. There are so many types of centrifuge we can find anywhere in the laboratory, but basically they have two different types, which is the gravitational force type and the centrifugal force type. When we are talking about the rotor or motor inside the centrifuge, so the centrifuge has two types of common motor centrifuge. The first is the swing out motor in the right picture here, and the second one is an angle fixed motor centrifuge on the left picture here. So the difference between two is that the first, the swing out motor, it has a tube pocket that can that stick to the rotor that can move freely. When the centrifuge motor rotates, the tube inside the centrifuge will be in a horizontal orientation due to the centrifugal force. And the second one is a fixed motor or angle fixed motor that the tubes unable or cannot spin freely inside the machine, and it's due to the gravitational force. The advantage of using this type of centrifuge is that the speed of the motor is faster, since there is a little to or no air friction in a centrifuge machine. So if we see here before and after centrifugation. So before the centrifugation process, the cell homogeneity is spread all over the tubes. But after the centrifugation, there is two different part of the tubes. So the pellet that these tubes contains two layer here. The supernatant that is always in the first layer, that because it contains a smaller and less dense component, and then the pellet one is the larger and more dense component. So the next one, we're gonna talk about the unit symbolize the force of the machine to separate the solution. The centrifugal force, or stated as G, which is affected by the motor speed and radial distance, or R, of the particle from the rotation axis. The G unit can be converted into RPM, or rotation per minute, and converted into RCF, or relative centrifugal force. According to its speed, there are different centrifuge as follows. The first one is microcentrifuge, which has a maximum speed of 12,000 RPM. That's why it's called microcentrifuge. And the second one is a high velocity centrifuge, which has a maximum speed of, for the microcentrifuge one is 25,000 RPM and a centrifuge force of 60,000 G. And the third one is ultracentrifuge, has a maximum speed of 80,000 RPM. So the ultracentrifuge is way faster than the other two. And when you are using the centrifuge machine, there are several considerations upon to use that machine. The first one is the centrifuge machine will operate when the lid is totally closed and the tubes are aligned and balanced. So it is necessary to weight the tube before putting it into the machine to minimize turbulence to meet the balance requirement. because unbalanced condition will enhance the turbulence and disrupt the sedimentation of the pellet that causes it to undo. So before you use the machine, it's better for you to weight your sample and make it balance between one and the second sample. If you only have one sample, just make sure to create the balance by using any liquid, for example using water or something. And this consideration number 2, it is better to fill up the centrifuge tube to 3/4 of the tubes to prevent bursts that cause a catastrophe. So please don't fill up until full. And the third consideration is make sure that the motor has completely stopped before opening the lid, because this is totally dangerous. And if you are in a laboratorium for the experiment, you need to do this experiment on the lab using 2 different samples here, using the centrifuge upon these 2 different speed, and you can tell the supernatant or pellet observation, like the volume of the supernatant and how many pellet you got. So moving on to the last part is SDS-PAGE Electroporator. So SDS-PAGE, or stand for the sodium dodecyl sulfate polyacrylamide gel electrophoresis, is a technique of protein separation based on their molecular weight. So SDS-PAGE is a technique of protein separation by their molecular weight. This technique is widely used in forensic, genetics, biotechnology, and molecular biology to separate the protein molecules based on their electrophoretic mobility. The principle of SDS-PAGE states that a charged molecule migrates to the electrode with the opposite sign when placed in electrical field. So you can just imagine that the molecule is migrate. And the smaller molecule migrate faster and the smaller molecules migrate faster due to less resistance during electrophoresis. So we can see here on the picture there are several parts of the SDS-PAGE electroporator. The first one is a casting stand here, like a white plastic here, the white transparent plastic here. Casting stand is used for the gel mold stand. So you're gonna put some gel and stand the gel on this casting stand. The second part is the casting frame here, and a plate glass like a sandwich. So there is a glass, two type of glass that you stick it together, and between the glass you can put the SDS solution here, and then the casting frame will press it and put it in a casting stand. So we're going to explain it later. And the second part, the third part is a plastic comb here. Is a plastic to make a well before a sample. So it's like, yes, it's like a comb, looks like a comb here. And then the fourth part is a glass plate with integrated spacer here, a glass to make an SDS gel, and the next one is a sample loading guide. Here is a guide for loading the sample, so you can just put your sample by using that sample loading guides. And then the last one is a clamping frame here and electrode assembling here. It's a box for the gel and its electrode. After you set up with the plate, you can put it here with the sample loading guides and put this one inside this chamber. So I'm going to explain you a little bit detail here. So the first, before you are making the gel, just clean the apparatus with the 70% ethanol in a picture. And a B, put the glass into the casting frame like this. Put the two different glass into this casting frame. And the C, lock the glass in the casting frame. Lock the glass in the casting frame, and put a casting frame in a casting stand there, and do a leak test using water, because if it's leaking, then you cannot make any SDS gel, because the liquid will be run over the glass, yes. And then the next one is take an acrylamide solution using a micropipette. Drip the solution starting from the resolving gel, the bottom one, and up to the 8 centimeter, for example, and until the staking gel, the upper part of the acrylamide solution or the SDS gel. And lastly, place the comb. So the comb is the last part, and wait for the gel to polymerize. And the B picture, the next picture, is install the glass plate that has the gel to the clamping frame and electrode assemble. And then the next step is pour the running buffer and get the comb. Then clean off the well using the micropipette. So it means that after the electrophoresis gel is set up, and put it in chamber, and take out the comb, and then clean the well using micropipette until no bubble. And the last one, pipette each sample one by one into the wall, and it's totally done. Okay, thank you for listening. Stay healthy and see you.